Hi dears, welcome back to Easy Classes by Soumya. Today we have to discuss about one of the important topic in biology, applied biology, biotechnology, principles and process. Okay, from any point of view, I am talking about CBSE, I'm talking about NEET exams, ISC exam, any other board exams, this chapter is very important. Also you have to discuss about or let you know about the emerging field of applied biology, biotechnology. Okay, let's start with what's meant by biotechnology. All of you know that the process like fermentation, making curd, bread, wine, etc. This field is coming under the old biotechnology. What's meant by biotechnology? Biotechnology is the use of living organisms or enzymes from the organisms to produce products that are useful to human beings. Okay, what's meant by biotechnology? Biotechnology is the use of living organisms like bacteria, fungi, any other cell of living organisms or enzymes from the organisms to produce product and services that are useful to human beings. Okay, let you know about the fermentation. By fermentation we we produces the appams. Okay, idli, dosha, etc. And also by fermentation we produces wine in our homes. Okay, so using microorganisms like bacteria, lactobacillus, yeast, unicellular fungi yeast, we can produce the products that are useful to human beings. Okay, this making of curd, wine, bread, etc. are come under the old biotechnology. Okay, then what's meant by the modern biotechnology? Modern biotechnology is also deals with the use of living organisms or enzyme from the organisms for products and services that are useful to human being. Here there is some kind of reconstruction or modification or manipulation takes place in the genetic material of these organisms. That's the difference between the old biotechnology and modern biotechnology. Nowadays, it is an emerging field of applied biology. There are many techniques are available to produce a desirable organism or to produce a desirable products, vaccines, antibiotics, etc. These are possible through technology in living organism, modern technology in living organism, the reconstructions in the genetic material, DNA, etc. Okay, so modern biotechnology deals with the synthesis of a defect, synthesis of a new gene, synthesis of a vaccine, antibiotics, and the IVF program, test tube baby program or in vitro fertilization, etc. Okay, let you know. Now I think you know well about the difference between the old biotechnology and the modern biotechnology. In old biotechnology we use only the microorganisms, the natural microorganism to produce products and services that are useful to human beings. But in modern biotechnology we use and the reconstructions, modifications or manipulations using the modern techniques in genetic material and new desired organisms, or plants or animals, or vaccines, antibiotics, etc. are produced. That's the main difference between the old biotechnology and modern biotechnology. All of you know about what's meant by biotechnology, modern biotechnology, and also the old biotechnology. Okay. Now we have to study the correct definition of biotechnology. Okay, what's meant by biotechnology? Simple definition. All of you know about the simple definition about the biotechnology, that according to EFB there is a definition for biotechnology also. Now first we have to study about what's meant by biotechnology. There's a simple definition. Okay, what's meant by biotechnology? Biotechnology is the technique of using living organisms or enzymes from organisms to produce products and services that are useful to human beings. It is the techniques of using organisms or their enzymes from the organisms or their enzymes for product and services that are useful to human beings. Okay, so that is the very simple definition of biotechnology. Okay, now all of you know about the who is the father of biotechnology. Biotechnology is a emerging field of applied biology. So we have to know about who is the father of biotechnology. Karoli Erecki is known as the father of biotechnology. Who is the father of biotechnology? Karoli Erecki is the father of biotechnology. Okay, let's start with the definition. The definition of EFB. What's meant by EFB? EFB means European Federation of Biotechnology. According to EFB, the definition for biotechnology is the integration of natural science and organisms, integration of biology and an organism, then the cells of that organisms and the parts or organelles of that cells like nucleus and its part chromosomes, DNA, etc. and molecular analogues means DNA or RNA. For what purpose? Product and services that are useful to human being. So we have to study the correct definition of EFB. There is no change in any word or sentences in this definition of EFB. Okay, what's meant by EFB? What's meant by biotechnology according to EFB? According to EFB, biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for product and services. Okay, that's all about the biotechnology. Okay, what's meant by biotechnology? Biotechnology is the technique of using living organisms or enzymes from the organism for product and process or services that are useful to human beings. Clear? Okay, according to EFB, what's meant by biotechnology? Biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for product and services. So, is the father of biotechnology. Karoli Erecki is the father of biotechnology. Okay. So, now we have to discuss about the two branches of biotechnology. The old biotechnology deals with the microbe-mediated process like making of curd, bread, wine, etc. The modern biotechnology deals with the in vitro fertilization or IVF or test tube baby program, and the second main important branch or important feature of the modern biotechnology is the synthesis and using of genes. Synthesing a correct gene അല്ലെങ്കിൽ correcting a defective gene or synthesizing a or using a desired gene, etc. are come under the modern biotechnology. And preparation of another feature is preparation of DNA wax and correcting a defective genes. Okay, old biotechnology deals with the micro-mediated process like making curd, wine, and bread. The modern biotechnology deals with the in vitro fertilization or test tube baby program, synthesizing and using of a genes, correcting a defective gene, and preparation of DNA vaccine, antibiotics, etc. All these are deals with the modern biotechnology. Okay, now we have to know about principles of biotechnology and processes involved in biotechnology. Okay, what's meant by principles of biotechnology? There are two main principles are involved in biotechnology. These are genetic engineering and bioprocess engineering. The first principle is genetic engineering. Second principle is bioprocess engineering. What's meant by genetic engineering? Okay, genetic engineering is the technique to alter the genetic makeup of an organism. What's meant by genetic engineering? It is the technique to alter the genetic makeup of an organism and introduce these into host organism and thus changes the external feature or phenotype of that organism. Okay, clear? Genetic engineering means engineering in the genetic material DNA or RNA. Okay, simply we have to tell that genetic engineering is the technology in the genetic material. So the correct definition for genetic engineering is the technique to alter, changes the genetic makeup of an organism, means DNA and RNA. First we have to make some changes in the genetic material, alteration in the genetic material DNA, and introduce this DNA into some host organism, and changed DNA is introduced into the host. Thus we can make the changes in the phenotype of that host. Okay, that is the definition for correct definition for genetic engineering. What is meant by genetic engineering? Genetic engineering is the technique to alter the genetic makeup of an organism and introduce these into host organism, thus change the phenotype of an organism. Okay, clear? We make some changes in DNA and introduce this DNA into a host cell and thus change the host organism, cell of the host organism, and make changes in the phenotype of that organism. That's all about the genetic engineering. Second principles of biotechnology is the bioprocess engineering. We have to maintain a sterile ambience or sterile condition for this genetic engineering process. In this process, genetic engineering process, we required only a desired microbes, only a particular microbes. All of you know that the atmosphere contains number of microorganisms, but we have to maintain a sterile ambience in genetic engineering process to promote the growth of only desired microbes. Okay, in large quantities. This is the second principles of biotechnology, bioprocess engineering. What's meant by bioprocess engineering? Maintenance of sterile ambience in chemical engineering process. Chemical engineering process means the alteration process or manipulation process or cutting process in DNA. For what? To promote the growth of only desired microbes. And to avoid all the undesired microorganisms in large quantities for the manufacture of antibiotics, vaccines and enzymes. We have to produce a particular vaccine for COVID-19. So we have to produces the vaccine or antibiotic that kill the COVID-19 vaccine only. Okay, so for this process, maintenance of sterile ambience in this chemical engineering process or genetic engineering process is required. For what? To promote the growth of only desired microbes in large quantity. You have to promote only the desired microbes. There is no need of another microbes. We have to produce large quantities of disease microbes for the production of vaccines, DNA vaccine, antibiotics, etc. So we have to maintain a sterile ambience. That's the second principles of biotechnology, bioprocess engineering. So bioprocess engineering means maintenance of sterile ambience in chemical engineering process to promote the growth of only desired microbes in large quantities for the manufacture of antibiotics, DNA vaccine, for correcting a gene, etc., etc. Clear? That's all about the principles of biotechnology. So now we already discussed about the topics: what's meant by biotechnology, old biotechnology, modern biotechnology, father of biotechnology, definition for biotechnology, simple definition, and also the unique definition of biotechnology by EFB, European Federation of Biotechnology, and also the principles of biotechnology, genetic engineering, and bioprocess engineering. So we have to recall these points once more. Okay, what's meant by biotechnology? Biotechnology is the use of living organisms or enzymes from the organisms to produce products and services that are useful to human beings. According to EFP, biotechnology is the integration of natural science and organisms, parts thereof, and molecular analogs for product and services. Okay, two branches of biotechnology. Old biotechnology deals with the synthesis of microbe-mediated process like the synthesis of curd, vinegar, bread, wine, etc. The modern biotechnology deals with the synthesizing a gene, correcting a defective gene, synthesizing enzyme, antibiotics, vaccine, and also the in vitro fertilization program, IVF or test tube baby program. Okay, next, the principles of biotechnology. There are two principles are involved in biotechnology: genetic engineering and the bioprocess engineering. What's meant by genetic engineering? Genetic engineering is the technique to alter the genetic makeup of an organism and introduce these into host organisms, thus change the phenotype of that host organism. Okay, we have to make some changes in the genetic material DNA and introduce these reconstructed DNA, modified DNA, or manipulated DNA, desired DNA into the host cell and produce large quantities of desired DNA. Okay, that's the definition for genetic engineering. The next principle is bioprocess engineering: maintenance of sterile ambience in chemical engineering process to promote the growth of only desired microbes in large quantities. Okay, what's meant by biotechnology? Bioprocess engineering: maintenance of sterile ambience in chemical engineering process to promote the growth of only desired microbes in large quantities for the manufacture of antibiotics, vaccines, etc. Okay, next we have to discuss about the first rDNA technology by Stanley Cohen and Herbert Boyer. Stanley Cohen, that is known as Stanley Cohen experiment. First rDNA technology was discovered by Stanley Cohen and Herbert Boyer. So this experiment is known as Cohen and Boyer experiment. Boyer and Cohen experiment, or Cohen and Boyer experiment. Okay, first rDNA technology is known as Cohen and Boyer experiment. Cohen and Boyer experiment. Stanley Cohen and Herbert Boyer experiment. This experiment, first rDNA technology, was discovered in 1972. Okay, let's start with the first rDNA technology. Okay, first let us begin with the rDNA technology. First rDNA technology, we have to know about some more topics, some more points, or some more things. Okay, we have to produce, our body wants some characters, some desired character that we that, that we that we quest for. Okay, the desired characters like the eyes of Aishwarya Rai, the height of Amitabh Bachchan, the intelligence of Einstein. Is it is possible to add in our own body or our own characters? Is it possible to modify our behavior, modify our appearance, external appearance? Yes, it is possible through the emerging branch of biotechnology, emerging branch of biology, biotechnology. Okay, how is it possible? Okay, we have to first we have to select a desired gene. This desired gene we selected from a donor. Okay, first step of our DNA technology is the isolation of donor DNA. Okay, then we have to, this is the donor DNA fragment, so we have to cut a small piece of desired DNA that is responsible for the production of that desired character. This is the, for example, this is the gene, this is the portion of DNA, or this is the portion of DNA, or this is the gene which is responsible for the production of, which is responsible for the expression of the feature blue eye color, or intelligence of Einstein, or the height of Amitabh Bachchan. Okay, so first we have to isolate the donor DNA, then we have to cut the desired gene from the donor DNA. This is possible through an enzyme which is known as molecular scissor, that is restriction endonuclease. Okay, then this this portion of desired DNA cannot transfer to the host cell alone. The desired DNA requires a vehicle or carrier for its transportation from donor to host cell. So a carrier or vehicle is required. The carrier or vehicle is known as vector DNA. The commonly used vector is plasmid vector. What is meant by plasmid? Plasmid is the extra, if it is a E. coli bacteria. All of you know that E. coli are an example. E. coli is an example for prokaryotic organism. Prokaryotic organism have no true nucleus, but they possess genetic material which are known as nucleoid. Nucleus, not nucleus, nucleoid. Okay, and it also possesses extra chromosomal circular DNA which is autonomously replicated within the host cell. Okay, this is known as plasmid. This plasmid is used as a carrier or vector to transfer the desired DNA to the host cell. Okay, so this is the vector. So we have to incorporate or insert this desired DNA to the vector. To incorporate or recombine the desired DNA to this vector DNA plasmid, we have to make a restriction or cut in the plasmid also to insert the desired DNA. So there is a small niches made in the vector DNA or plasmid DNA also with the help of enzyme restriction endonuclease. Small portion is removed from here. Okay, small portion of DNA is also removed with the help of restriction endonuclease. Now we get a desired DNA. And also we have a plasmid DNA. Okay, we this is desired DNA, foreign DNA or aligned DNA. This is plasmid or vector DNA. Okay, the next step is the insertion of the desired DNA into the plasmid vector with the help of an enzyme which is known as molecular scissor or DNA ligase. With the help of DNA ligase, this desired DNA is inserted into the vector DNA. So we get a reconstructed or recombined DNA. Then this plasmid is known as recombinant DNA. Okay. Next we have to transfer this recombinant DNA to a host cell bacterium. Host cell is the bacterium. So we transfer this recombinant DNA into the bacterial cell. So the bacteria have the ability to or replicate very quickly and also to produce their offsprings in very quick time. Okay, its replication, the time required for the replication of bacteria is only 18 to 20 minutes. So it produces a large amount of rDNA within very few duration or time. Okay, thus we get large amount of desired DNA. This desired DNA with the plasmid is cultured in a culture medium and it produces large amount of antibiotics or vaccine, etc. Or the desired gene, like the gene of Ishar, which is responsible for blue color, the intelligence of Einstein, etc. Clear? That's all about the rDNA technology. So first we have to discuss about the first rDNA technology. Okay, first rDNA technology is proposed by or discovered by who? ആരൊക്കെയാണ്? Discovered by two scientists, Stanley Cohen and Herbert Boyer. Stanley Cohen and Herbert Boyer is the two scientists who discovered first rDNA technology. Okay, let us know about the first rDNA technology. Okay, here they selected the desired gene. The desired gene or desired DNA is the desired DNA selected for rDNA technology is the antibiotic resistant gene. This antibiotic resistant gene have the ability to produce the antibiotics. Okay, and antibiotic resistant gene is the desired gene. This antibiotic desired gene is selected from or isolated from a donor DNA. The donor is the plasmid of Salmonella typhimurium. So the donor DNA is plasmid of Salmonella typhimurium is the donor DNA. From the donor DNA we cut a small portion of desired DNA with the help of enzyme restriction endonuclease. Okay, so this is the desired DNA. The plasmid of Salmonella typhimurium possesses number of antibiotic resistant. A small of antibiotic resistant gene is cutting with the help of enzyme restriction. So this is the antibiotic resistant gene. And we have to transfer the desired gene into host cell. Bacterium produces large quantities of antibiotic resistant gene. So this antibiotic donor DNA or desired DNA does not have the ability to transfer to the host. Okay, so we required a carrier or vehicle. The carrier is plasmid vector. Okay, the carrier used here is a plasmid vector. So we make a restrictions or nick in the plasmid with the help of restriction endonuclease, same restriction endonuclease, and a small portion is removed, and this is the plasmid after this is the remaining plasmid vector DNA after restriction. And the next step is the insertion or recombination of this desired DNA into the plasmid vector or the carrier vector, and we reconstructed a recombinant DNA. Okay. So now you have to produce a large quantity of recombinant DNA. For this process, we transfer the recombinant DNA into a culture medium where bacterium is growing. Bacteria have a culture medium for its growth, okay, for its replication or multiplication. The agar and the sucrose, lactose containing medium. The bacteria are grown in a culture medium. In this culture medium, we also put this recombinant DNA. So the bacteria have the ability to take up the foreign DNA. So the bacteria take up the recombinant DNA. The bacteria which take up recombinant DNA are known as transformant bacteria. The transformant bacteria with the recombinant DNA get the ability to have the ability to multiplicate or replicate within very few hours, and they produces large quantities of recombinant DNA. So from this recombinant DNA we can produce as antibiotic resistant gene. Clear? That's all. First rDNA technology by Stanley Cohen and Herbert Boyer. Okay, the first rDNA technology is proposed by Stanley Cohen and Herbert Boyer in 1972. Here the donor DNA is this Salmonella typhi, the plasmid of Salmonella typhimurium. First donor of the donor of the first rDNA technology is who is the donor? Plasmid of Salmonella typhimurium. Okay, then from the donor DNA, small portion is cut or restricted with the help of restriction and enzyme or restriction endonuclease like E. coli, BAMH1, etc. Okay, then that portion is isolated. After that, we select a vector or carrier or vehicle for transporting or for transfer of the desired DNA to the host cell for multiplication. Okay, the plasmid, we use the plasmid vector as a vector. So plasmid is an extra chromosomal circular DNA which have the ability to autonomously replicating within the host cell. When this plasmid enters another bacterium or another host cell, this plasmid have the ability to replicate within the host cell autonomously and produce younger ones. So a small portion is also cut with the help of restriction, same restriction endonuclease from the complementary counterpart. A small portion means complementary to the donor DNA. Okay, and the selected desired DNA after restriction is also inserted into the plasmid vector, plasmid DNA fragments with the help of enzyme, the molecular gum, the enzyme which is known as molecular gum DNA ligase, and we get a rDNA. Then we put this rDNA into bacteria's culture medium. The bacteria take up the recombinant DNA and they undergo multiplication or replication, produces large quantities of recombinant DNA. Okay, clear? That's all about the first rDNA technology. Okay, in first rDNA technology, the donor DNA is the plasmid of Salmonella typhimurium. Very important question for board exam and important MCQ for board exam and NEET exams. Okay, the first donor DNA is the plasmid of Salmonella typhimurium, and a small portion is cut from the plasmid vector, cut from the plasmid of Salmonella. The desired gene is antibiotic resistant gene. Then we select the plasmid vector. A small portion is also cut from the plasmid vector to enter the desired DNA to plasmid vector, the complementary part. The complementary to the desired DNA, small portion which are complementary to desired DNA is cut from the donor DNA. And these two DNA fragments are ligated or linked with the help of enzyme DNA ligase, then RDNA is formed. Okay, this is the first RDNA technology. Okay, I think all of you know very well about this topic now. Okay, now we have to know about the next part of this chapter, tools involved in RDNA technology. Which are the tools involved in RDNA technology? First first to start this process, first to produce a useful product from living organism, we have a donor that provide the desired DNA or alien DNA or foreign DNA. So first we isolate the donor DNA. If donor DNA may be a plant cell, animal cell, bacterial cell, or cell of antibiotic resistant cell of microorganism, etc. Then the small portion, the small portion of desired DNA or desired gene is cut with the help of restriction endonuclease. Okay, so the another tool, restriction endonuclease, is used to make restrictions in the donor and vector DNA. Then these donor DNA fragments and vector DNA fragments are linked together with the help of the enzyme which is known as DNA ligase. Then we get a recombinant DNA. These recombinant DNA are then now we have to produce a large quantity of vaccines or antibiotics. So for the large scale production or the production of a huge quantity of the product, we have to transfer this recombinant DNA into the culture medium of the bacterium, where the replication of the recombinant DNA or bacterial DNA is happen. Thus we get large quantities of desired DNA, and from this desired DNA we have to produce DNA, vaccine, antibiotics, etc. Okay, so which are the tools involved in RDNA technology? We must have a donor DNA, but we does not mention here donor DNA as a tool in this chapter. So in NCERT textbook, the first tool is the restriction endonuclease enzyme, which is known as molecular scissor. Okay, the second tool is vector DNA. Vector is the carrier or vehicle which have the ability to transfer desired DNA to host cell, recombinant DNA to host cell, and produces large quantities of recombinant DNA. Then we require a DNA ligase enzyme to recombine the donor DNA with the vector DNA. Next, last tool is the competent host. To produce multiple copies or large amount of the donor, we have a host. The host here used in RDNA technology or biotechnology is bacterium. Competent host is bacterium. Okay, that's all about the tools. Which are the tools involved in donor DNA, vector DNA, or restriction endonuclease, molecular scissor, vector DNA, plasmid, extrachromosomal circular DNA, which has the ability to autonomously replicate within the host DNA, and enzyme which is known as molecular gum DNA ligase, and a competent host. The host is known as bacterium. The host is normally we used here the microorganism bacterium as a host, competent host. Okay, that's all about the tools involved in RDNA technology. Next, we have to discuss about the first tools involved in RDNA technology. First tool is Which is the first tool? Restriction endonuclease, which make restrictions within the donor DNA and vector DNA. Okay. Second tool is the DNA ligase. Of course, DNA ligase. And the third tool is plasmid vector. Plasmid vector means extra chromosomal circular DNA of bacterium, which have the ability to autonomously replicate within the host cell. Okay. Next tool is the competent host. Host must be competent to take up the DNA. Since DNA is a hydrophilic substance which does not have the ability to take up the natural plasmid. So we have to make the host bacterium competent. The bacterium undergo process which is known as heat shock method or electroporation. Okay, let's start with the first tool involved in DNA technology is restriction endonuclease. Okay, clear?